Hi, welcome to Science Hangout. So we have today Dr. Kristen Brennand from Icahn School of Medicine at Mount Sinai, New York with us. She is assistant professor in neuroscience and psychiatry. Dr. Brennand is a stem cell biologist working to bridge the fields of developmental neuroscience and psychiatry. A major focus of her work is developing in vitro models for schizophrenia in order to identify novel insights into the molecular and cellular phenotypes of mental illness. In 2013, Dr. Brennand won NYSCF Robertson Investigator Award. Also in 2013, she won NIH Behavioral Research Award for Innovative Neuroscientists. Dr. Brennand has published several articles in prestigious journals in last several years. Welcome, Dr. Brennand, to Science Hangout.
Hi, thank you. Thank you for inviting me.
Can you please talk about yourself and your education research projects you are working on currently?
Sure. So I guess what you probably are asking is I did my undergraduate training in Calgary, Canada. And then I moved to Boston to be a graduate student at Harvard University in the lab of Douglas Melton. And finally, I moved to San Diego, California to do postdoctoral training with Fred Gage at the Salk Institute.
Okay.
So with Doug, I became a stem cell biologist. And then later in California with Rusty Gage, I learned to apply stem cells towards neuroscience questions. And so that's what I've been doing since I came to the Icahn School of Medicine in New York about 4 years ago, is using stem cells to answer questions in psychiatry.
Okay. And in your short, brief career as an independent investigator, so can you please talk about some of the role models or mentors you have come across?
I mean, I think I've been really fortunate to work in the labs of some amazing scientists. Doug, my PhD advisor, he was a brilliant scientist, but also he cared so much about the people in his lab. And so when I really valued the mentorship that I got from him. And so when I chose my lab to do a postdoc in, I think I got just as lucky working with Rusty Gage, who again has a lab full of great postdocs who all work really well together under his guidance. And so from him, I also learned the value of managing a team. In his lab, I think it's always true that the sum of the parts is much greater than it should be. And I think that's because of the way he runs the lab. And so I'm really lucky to work in two fantastic labs in my training.
Okay. And what motivates you every day to work in this field?
I think it's important to ask questions that or to have questions that drive you. And for me, neuroscience is just full of mysteries. We know so little about the brain sometimes, and to have a tool, in my case, stem cells, that we can use to ask questions in a simple but new way. I think it makes it really fun, since we come in every day and I don't know what I'll learn each day, and I think that's what drives me.
Okay. And as an investigator, what are the challenges do you face every day?
Well, I think that there are two types of challenges. When you are a grad student or a postdoc, the challenge is learning how to design an experiment, how to interpret it. Day to day, I think that's the struggle. And now, as you rise through the career, I'm less and less at the bench. And so the challenge is no longer how do I design an experiment, but how do I teach a student how to design an experiment? How do I help a postdoc anticipate what will go wrong in their experiments before it goes wrong? And so learning to mentor people is my greatest challenge these days, but it's a fun one.
And how do you motivate your grad students and postdocs?
I really stress the lab as a team. I truly believe that there are no lone wolves in a stem cell lab. Everybody needs everybody else. Our projects are independent, but mutually beneficial. Any assay, any cohort, any differentiation protocol established by one person in the lab can be immediately utilized by others. And so I try to only hire team players, and we stress all the time that we're working on everything together, that your project is 80% yours and 20% going to be finished by work done by others in the group.
Okay, that is a nice way of getting things done, I guess. And can you please talk more about your lab people?
So again, not just in my lab, but I think science in general is a team exercise. And so I have 4 postdocs in the lab right now, but 2 of them are actually co-mentored by other larger labs in the Institute. And of my 3 grad students, one of them is also co-mentored by someone else at the Institute. And so it allows us to bring knowledge and skill sets and methods and analysis strategies developed in other labs into ours. And I think it's the most fun way to do science, is when everybody is bringing different expertise into the same room.
Okay, and let's talk something on research your lab is working on. So can you please highlight some of the research going on, and whenever you get a chance, you can also present your work?
Yeah, so really briefly, I think one of the biggest problems in psychiatry over 50 years of research has been a lack of access to patient material. So in cancer, for example, the patients are paying you, begging you to cut the cancers out of them so that you have them to study. But in psychiatry, it's not the same. You can't just go into a patient's brain to take samples. You can't go into a healthy control's brain for a point of comparison. And because of this lack of access to tissue, it's been very hard to answer fundamental questions in psychiatry. We don't know in what cell type things go wrong first. We don't know what cell type we should be targeting for therapeutics. And I think that's something that I want to address in my lab. Really briefly, we've built upon the discovery made by Shinya Yamanaka in 2006 that anyone's skin samples can be reprogrammed to stem cells. We call them induced pluripotent stem cells. And those stem cells can then in turn be directed to differentiate into any cell type in the body. So by standing on the shoulders of giants, of discoveries like this, we are able to take skin samples from patients and controls, and then ultimately turn those into neurons or astrocytes, the major cell types in the brain, to ask really simple questions. In what cell types do we see cell autonomous deficits in schizophrenia? What molecular changes drive those deficits? And ultimately, can we screen for drugs to make them the same again?
Okay. And if you get a chance, you can present your work so that it will help us understand more on what you are doing.
Sure. I'm really excited today to present some work that actually was only published in Cell Reports about 2 months ago. So this is really new data that I'm excited to share with your group today.
Okay, thank you.
So unfortunately, I don't think the animations are going to work on this platform, but hopefully if your audience will bear with me, we'll get through it okay. And so I'll be talking today about modeling predisposition to schizophrenia using induced pluripotent stem cells. So first of all, for those who aren't familiar, schizophrenia is a very common and debilitating disorder. It affects about 1% of the people in every country of the world. That includes 3 million Americans today. There is no cure for this disease. And in fact, patients show an elevated risk for suicide. 1 in 10 will commit suicide. For substance abuse, 1 in 2 will have drug and alcohol problems. Unemployment, 9 out of 10 will not hold a full-time job after diagnosed. And homelessness, 1 in 5 will live on the streets. I'll also add that the estimates of heritability for this disease are extremely high. They range from 50 to 85%. But unlike other diseases, it's not a simple Mendelian disorder. So estimates are as many as 1,000 genes might be contributing to onset of schizophrenia. And so this is the main figure from the largest genetic study of schizophrenia that's been done to date. It looked at the DNA of more than 35,000 patients with schizophrenia, and it found 108 loci that were contributing to disease in these patients. And so So one of the things that I always like to stress is that this is a complex genetic disease. And in 50 years of studying it, I think this is one of the major things that we've come to understand, that it's not simple inheritance. And what else have we come to understand in 50 years of schizophrenia research? And I think it's summed up here in this slide, perhaps overly simplified, but the basic idea is that human neurons are less well connected in schizophrenia. And so on the left, you can see brain imaging scans. Red indicates brain regions with reduced cell volume. And so brain imaging scans and also post-mortem analysis indicate that the brains of patients are smaller. The catch here is that the brains of patients are not smaller because neurons are dying. There are not fewer neurons in patient brains. Rather, the neurons are smaller and they have fewer connections between each other. And this is illustrated in the picture on the right, where you can see reduced synaptic density in the patient, the bottom panel, relative to the controls. So that being said, if what we know about schizophrenia is that it's a complex genetic disease characterized by poorly connected neurons, what do we still not know? And there's a lot of really simple questions that we still don't know. The major question that we don't know are which neural cell types underlie the cellular deficits in schizophrenia. I can tell you that the only FDA-approved therapeutic for schizophrenia is a dopamine receptor antagonist. Mm-hmm. The mouse models imply a link between glutamatergic connectivity onto GABAergic inhibitory neurons. But there are many others who think the support cells, the astrocytes, the oligodendrocytes, and the microglia are also impacted in disease. And so the truth is that there is evidence that all 6 of these cell types are perturbed. But one of those simple questions that we don't know is which cell type goes wrong first.
Yep.
Another simple question that we don't know the answer to is when and how do those complex genetic risk factors interact to produce disease risk, and when and how do they interact to produce clinical onset? And so the truth is that of those 108 loci, they tend to be enriched for genes that are expressed in early field development. And so it seems like that some or many of these risk factors are genes that are going wrong before a person is born. There are others certainly that are important in childhood, and others and environmental factors too that contribute during adolescence. And so one of the things that I'm hoping to understand in the lab is predisposition to disease. So what is going wrong before the patient is born? What's going wrong before they show up in the clinic? And so why don't we have the answer to these simple questions? And as I mentioned before I began the talk, in my opinion, one of the reasons that we don't have the answers is that there's insufficient live human brain tissue for studies of schizophrenia. There have been mouse models of this disease for 20 or 30 years already, but it's important to understand that the mouse brain is not the same as the human brain. It's much less complex, and there are probably certain questions that can only be asked in human cells. And so how can we go about getting human cells from patients without requiring biopsies of their brain? And so I'd like to take a minute to define 3 words that I'll be using a lot in this talk. The first word that I'll use a lot is differentiation. So differentiation is directing a stem cell towards a fate like a neuron. It's falling down the developmental landscape from a more pluripotent state to a more defined state. It's the top term. Reprogramming in the middle is the opposite. It's converting a differentiated cell back up the landscape to a more undifferentiated state so that later you can differentiate it into something else. So an example here would be reprogramming a skin cell back into a stem cell so that ultimately you can bring it back into a neuron. And induction is more of a lateral transfer, this bottom term. It's converting one cell type, like a skin cell, directly into another, like a neuron, without going back to the source stem cell. So these are words that I'll use a lot today. And this is a schematic illustrating exactly what we're doing in the lab. So again, briefly, we have a patient with schizophrenia and a matched control. And we take skin samples from those patients. Those skin samples are reprogrammed into induced pluripotent stem cells and then differentiated first into these immature neural progenitor cells and ultimately into postmitotic functional neurons. So these progenitor cells, NPCs, are replicative and have the potential to give rise to both neurons and astrocytes. So this methodology is new. It wasn't even possible 10 years ago, and as a new technology, there are both pros and cons. It gives us the ability to produce limitless numbers of live human neural progenitor cells, NPCs, and neurons for the study of disease progression, things that go wrong before patients show up in the clinic. Hopefully, it will allow us to correlate the gene expression, the RNA expression levels, and cellular phenotypes in NPCs and neurons to those genetic loci that have already been identified in patients. And ultimately, we hope that this will serve as a high-throughput screening platform that might allow us on a genotype-dependent basis to identify new therapeutics for specific patient populations. The cons, as with any system that's new, we're still not that good at it. The NPCs and neurons that we generate are heterogeneous, and as a result, the neural networks that form are artificial, immature, and tend to lack myelination. So what does this look like in the lab? Well, I'm showing you here 3 sets of pictures. On the left are the stem cells, in the middle are the neural progenitor cells, and on the right are the neurons. I like to show this image to really point out that at this level of magnification, you don't see differences. There's no failure to make NPCs or neurons in these patients. And that's important because they're walking and talking and breathing like the rest of us for most of their life. And this was early work that was helped a lot by the 3 students in the picture back in San Diego. So the question then, before you can compare patient and control cells, is how closely do these stem cell-derived NPCs and neurons reflect what we see in the human brain? And to answer that question, we used a really great resource, the Allen Brainspan Atlas. We were able to compare gene expression profiles here in the top in the graph, in the control NPCs in black and neurons in white, to the postmortem tissue collected in the Allen Brainspan Atlas. What we found was that our NPCs, these black bars, are most similar to first trimester fetal tissue, while our neurons are most similar to first and second trimester fetal tissue. I think that was a good reminder because schizophrenia, which is all the way out here on the right, is obviously a disease much later than the age of our cells. And so we're not modeling schizophrenia in a dish. Rather, we're modeling predisposition to schizophrenia in a dish. On top of that temporal age, we also asked a spatial question. To what brain regions are our cells most similar? And here in the top panel of the controls, you can see that our stem cell-derived neurons most resemble the cortex and the subcortex. And in the patient-derived neurons, the same thing is true. Cortex and subcortical tissues are the best hit. And so here we have it. We really thought we had apples that we were comparing to apples, that there was no difference in the patterning of patient and control cells. So we could just go ahead and ask, how are they functionally different? And so the last thing that you want to do as you begin comparing patients and controls is to think really carefully about what types of patients and what types of controls. And so this early work, the patients were picked quite simply based on what was available. This was early times. There were not a lot of patients with fibroblast samples available. So the early reports you can see here in 2011, 2013, really reflected these cohorts of whatever was available. More recently, we and others have begun to move towards cohorts that either have a similarity in their genetics, they have a shared mutation, or a similarity in their clinical aspects. This has already been particularly well applied to bipolar and autism, and a direction that we're moving now with schizophrenia. And so I think it's very careful, one must be thoughtful that if you are going to take an extremely small patient cohort, when the geneticists are looking at 30,000 patients and we're looking at 5 or 10, we're very underpowered. So it's important to try to pick patients moving forward that are as similar to each other as possible. Similarly, you should equally agonize over which controls. And again, those older papers, we tended to use unrelated healthy controls, basically controls who were available. And more recently, it's been common to see people using unaffected relatives if they're looking at a particularly severe and penetrant mutation, or isogenic controls. This is when we can use CRISPR to precisely fix the mutation in a patient or mimic it in a control. And so this paper here by Wen et al. Is a really great example where they took advantage of both All three types: unrelated controls, unaffected relatives, and isogenic controls. So that being said, this is work that I began a long time ago. It's a cohort of whatever was available, 4 patients with no shared clinical or genetic characteristics. And we asked in them if we could model the neural deficits underlying schizophrenia in those replicative neural progenitor cells. And so one of the ways that we began was by unbiased gene expression analysis. First, I'll point you at the blue box. Here we're doing a weighted gene co-expression network analysis. And we're asking what types of genes are changing together in the patients. And the major network that's perturbed I've boxed in blue, and it includes genes involved in neuron differentiation and synaptic transmission. I'll remind you that this analysis was done in those replicative NPCs. They're not mature neurons. And so this analysis really suggested to us that the aberrant gene expression characteristic of schizophrenia in neurons existed in those NPCs prior to neuronal differentiation. And in yellow is the second most significant box, and it indicated perturbation of a neuronal migration network. So we were actually able to follow up on this migration network in cells because we were able to develop a number of assays for neural migration. Actually, I think I have an extra slide here. Here we go. And so a really great technician in my lab, Ngoc Tran, worked to develop a few assays of neural migration. In this first one, we call it a neurosphere migration assay. She asked just simply, how far do cells migrate from the original neurosphere? So cells were grown in suspension for a few days to generate neurospheres, and then Ngoc manually picked thousands of these neurospheres, waited 2 days, fixed them, and analyzed how the average radial migration of each sphere. And in the top graph, what you can see is the results, that there was a significant decrease of radial migration in the patient-derived NPCs. This was not a diagnostic difference, but it was on average true and very significant. And because this assay only took 48 hours, we were able to ask a number of really simple questions afterwards. The first question we asked was this due to secreted factors? And so we did a conditioned media exchange. So in this bottom panel here, what we've done is we've taken control neurospheres and cultured them either in control conditioned media or patient conditioned media. Similarly, the patient neurospheres were cultured in control or patient conditioned media. And we did the neurosphere analysis. Again, you can see that there is no effect contributed by the conditioned media. The patient cells just consistently migrated less far than controls. The next question we wanted to know is whether this was mediated by contact. And so now we've generated mixed neurospheres. In the top panel, the patient cells are green and the controls are red. And in the bottom, the patients are red and the controls are green. And in each case, we ask which cells migrated further. And pretty consistently, what we learned was that it didn't matter whether you labeled the patient cells green or red, they just migrated less far than the controls. And so this told us that this difference in migration, it wasn't due to a secreted factor, it wasn't due to a contact cell adhesion molecule, it seemed to be due to something intrinsically cell autonomously inside the cells. We followed this up with a couple of extra migration assays just to confirm these findings. So on the left is that neurosphere migration assay. In the middle is a microfluidic device assay. Here we plate the cells in the bottom chamber in A and ask how long it takes them to move through the microfluidic device to a top chamber in B. And so they're going from this bottom to this top. We give them 5 days and we simply count how many cells move. And again, you can see a significant decrease in migration in the patient NPCs. And this third assay is a hydrophobic surface assay. Cells are spotted on spots of laminin. So originally there are only these spots, these circles. And 3 days later, after 3 days, they begin to crawl over each other to connect the dots. And you can see a significant failure in these patient-derived NPCs to crawl over each other to connect the dots. And so with this number of assays, we really felt confident that we had a migration deficit or aberrant migration. And the question that we wanted to ask was, first, could we identify the underlying molecular factors contributing to aberrant migration with the thought that these might represent risk factors for schizophrenia predisposition? So we move forward to RNA-seq and microRNA experiments to ask which microRNAs and which genes were different in the patient NPCs. And so Here, what we've done, one of the first experiments actually when we started the lab was we unbiased characterized expression of 800 human microRNAs across our 4 patients and 6 controls. We learned 2 really interesting things in this analysis. First, we learned that microRNA 9 was one of the most abundantly expressed microRNAs in NPCs. And second, we learned that actually microRNA 9 was one of the most perturbed microRNAs in patient NPCs. I also, a quick literature review revealed that microRNA 9 was involved in neural differentiation and neural migration. So it promptly became one of my favorite microRNAs. So here on the left graph is the nanostring data showing decreased expression, again, not diagnostic, but significant on average in the patient NPCs, and a qPCR validation that this microRNA was indeed changing. At the same time that I was doing this microRNA analysis, a collaborator of mine, Gong Fang, was doing an integrated RNA-seq analysis. So what Gong was doing was he looked at global gene expression, and because we know which genes are targets of which microRNAs, he was able to predict from those genes that are changing which microRNAs might be driving it. And you can see that he independently from a different data set from the RNA-seq data also identified miR9. So this is on the left. That's this top most important microRNA regulator. Then we integrated my analysis here on the y-axis, microRNA expression, with his analysis, microRNA activity, which is defined by the changes in the target genes. And what we got was a really significant result. It's probably, in fact, the best R value I will see in my entire career. R of 0.97, really significant, p to the negative 7. And so you might ask, well, how meaningful is this? Of course microRNA expression and activity should be correlated. But it turns out of all the other microRNAs that he identified as having target genes that were changing, there was not such a strong correlation between expression and activity. In fact, the only one that had this significant strong association was microRNA 9. And so that's the one that we followed up on in the study. I'll also add that this correlation between microRNA expression and activity of miR9 was unique in the NPCs. So on the left is a graph showing miR9 expression and control in gray and patients in red in NPCs through differentiation to 6-week-old neurons. Then you can see that by the time we've made mature neurons, we've actually lost that differential expression. Moreover, when you compare microRNA expression and microRNA activity in the neurons, now we've got one of the worst R values of my career, R of 0.04, P of 0.92. It's not significant at all. And so we really then focused on miR9 expression differences in patient NPCs. One of the first things that a really great technician of mine in the lab, Aaron Topol, helped me ask was whether we could change or ameliorate this migration deficit by overexpressing miR9. So in the left panel, you see control and patient-derived NPCs overexpressing just GFP. And again, you can see decreased migration in the patient NPCs. But here we've overexpressed miR9 in these cells as well as GFP. And what you can see in this graph is that overexpression of miR9 was sufficient to ameliorate aberrant migration. Moreover, knockdown of miR9 in controls was sufficient to perturb migration. So here we had some good evidence that on top of being one of the most perturbed microRNAs, direct manipulation of miR9 levels was sufficient to restore migration. We wanted to understand what the target genes were that were changing. And so we did a second RNA-seq experiment. This one we're calling miR9 perturbation. So here for 2 controls and 2 patients, we overexpressed miR9, we had baseline readings, and we knocked down miR9, and we asked what the global gene expression effects were. In this top graph here, what we've done is we've correlated miR9 expression targets with miR9 fold change matched in the RNA-seq data set. What we're able to show is that the gene expression fold change of the miR9 targets is very highly correlated to miR9 fold change. We can also look at the genes that are changing, and we see a significant enrichment, 1.53-fold, 10 to the minus 5, of miR9 targets in the data set. Moreover, there's 108 loci are enriched. The genes in those loci are enriched in our data set at 10 to the minus 4. And so miR9, genes that change when you inhibit miR9 have a strong enrichment for schizophrenia genes. Finally, because we were unable to find one single target, instead we saw evidence that lots of genes were changing, we decided to integrate our two RNA-seq data sets, this miR9 perturbation on the left and our NPC signature, where we compared case and control on the right. And when you look at what types of genes overlap between these two data sets, you can see two classes of genes changing. The first are direct miR9 targets. So these would be the genes that go down when miR9 is up and go up when miR9 is down. The significance here is very small. It's not significant at all, in fact. Whereas the indirect targets, those genes that go up when miR9 is up and down when miR9 is low, those are significantly enriched at 10 to the minus 20. So those are the genes that are in common between these two data sets. We look closer at what these shared indirect targets were. And there was 37 overlapping indirect targets. First of all, they again were enriched for known schizophrenia genes. And second of all, they were disproportionately represented by migration and adhesion genes. Oops. And so this is really what we knew from the study. We knew that miR9 levels were decreased in patients. miR9 target genes were enriched for changes. Manipulating miR9 ameliorated our migration phenotypes, and that the overlap between the genes that are changed when you manipulate miR9 when you compare case and control are actually a bunch of indirect miR9 targets involved in cell adhesion and migration. The last thing that we did in our study was to replicate it in a second cohort. So this first cohort I talked to you about, the 4 patients, they had schizophrenia, but our replication cohort is actually defined by childhood onset schizophrenia. So it's an earlier onset, more severe version. And so briefly, childhood onset schizophrenia is diagnosed between the ages of 6 and 12 by the same clinical criteria as used for adult onset schizophrenia. There are no unique genetic risk factors to childhood onset schizophrenia that are not observed in adult onset schizophrenia. And finally, in longitudinal studies, childhood onset patients are indistinguishable from chronic adult onset schizophrenia patients as adults. And so here now we have 2 data sets. This is our original cohort of 4 patients, 6 controls. This is our new cohort of 10 patients and 10 controls. And here is our replication cohort. And again, you can see a significant decrease in miR9 expression in the patients. Not diagnostic, but significant. And the last thing that I'll point out when we integrate these 2 data sets is that together, across both data sets, miR9 levels are decreased in patient NPCs to the extent that, first of all schizophrenia NPCs have miR9 levels less than the median levels in the controls. And specifically that this signal is driven by 50% of the patients with values less than the bottom quartile of controls. And the strongest effects are from the bottom quartile of patients. And so while on average is true, the signal is driven by a subset of patients. And so this is really the end of this specific IP story. But at the same time that we were doing these stem cell studies, the geneticists were moving ever onward. So I showed you this slide earlier, but they've been slowly accumulating more patients. And it turns out one of the SNPs that's just reaching genome-wide significance now is in miR9-2. And miR9-2 is the only microRNA 9 version of microRNA 9 that's expressed in the human brain. Not only is miR9 as a loci becoming significant for schizophrenia, but so are its target genes. And so miR137 to date has been the only microRNA whose targets are significantly enriched in schizophrenia GWAS. But now, in a recent paper published in March of this year, they've also shown that miR9, its targets are significantly enriched in the schizophrenia GWAS. And so here are 2 independent approaches, my stem cell-based approach and this genetic-based approach, converging on the same microRNA. And so I think this is a great proof of concept that the stem cell field will be useful in discovering and validating genes that are coming through in the schizophrenia GWAS. And so the most important slide that I can show to the members of my lab and my funding support and our collaborators, because this work, it doesn't occur in a vacuum. And without everyone in the lab helping to finish this project and others, it just wouldn't get done. And we have collaborators around the country helping us recruit patients, analyze cells and really understand the significance of the findings in the lab. And so I want to thank all of you for listening, and I'm happy to take any extra questions at this point.
Thank you so much. It's very interesting work.
Thank you. Thank you very much.
And I have a couple of questions.
Yeah.
Like you are doing those migration assays, right?
Mm-hmm.
So I was just wondering, like in the human patients, is there any migration problem with the neurons in the autopsy?
So that's a great question. I get asked it a lot. And the answer is that it's a hard one to know because you don't get good match controls. Patients die in different ways. The structure's not there. I would say in mouse models, there's a little bit of evidence that aberrant migration might be happening. Mm-hmm. But I think the more important parallel to draw is that those genes involved in neural migration, they're the very same ones involved in synapse formation. Okay. And so I might just be modeling the model. That what we're seeing in the dish is aberrant migration, but what happens in patient brains is poor synaptic connectivity. The same genes are contributing to both.
Oh, okay, that is interesting to know. And other question is regarding MIR9. Is there any other, like in other tissues, the role of MIR9 has been defined?
So MIR9 is involved not just in the brain, but the other, so there's 3 versions of MIR9 in the genome, and the other ones are expressed in other tissues. I think that they're typically involved in migration, things like epithelial to mesenchymal transitions.
Okay.
But I'm not sure which other diseases MIR9 has been implicated in, to be honest.
Okay, okay. Because I was trying to see if at all you can extrapolate in from other tissues anything, because what you are getting is kind of a wide variety, lots of genes getting affected. Mm-hmm. So yeah, it's an interesting presentation. So you don't think any other MIRs are involved currently?
Oh no, there are certainly many other microRNAs involved. This was just the first one that we were able to best demonstrate in the lab.
Okay, okay. And is there any work done on the regulation of these MIRs in the brain, like MIR9 specifically, or like how they are regulated in the brain?
So people have actually been looking at this for a long time, actually. And so it turns out some of the most critical genes that regulate neural progenitor cell fate in vivo, SOX2, TLX, they also regulate MIR9 and are regulated by MIR9. So there's a very complex interaction between the transcription factors that maintain these cells as neural stem cells and MIR9 in the brain. And so I think that's maybe why the cells are so exquisitely tuned to being sensitive to perturbations in MIR9 levels.
Okay. Looks like you have a lot of work you can do with this, because there are a lot of targets are really not specified and all those things, which is in fact good to have this kind of problem.
It's great when the questions outnumber the answers. Lots of work to be done.
Yeah, yeah. So again, I thank you very much for your time and effort in making us understand more about schizophrenia.
It was my pleasure. Thank you very much.
Thank you.